Hello everyone. I'm Siddharth Kulkarni. I am a PhD candidate at the George Washington University in Gustavo Hormigas lab, and today I'll be talking about the smallest orb-weaving spiders in the world. When we say spiders, generally, okay, my slides are not working. Okay, when we say spiders, generally we tend to imagine the orb weaving spiders, which is the iconic web that we know of. And some of us who are interested in other groups of spiders tend to think of their spiders of interest. But what about the smallest spiders in the world? So when I say the smallest spiders in the world, this spider known from Colombia, Patu digua, is just 0.37 millimeter long. For those of us who do field work and who may have been in that region, we may have inhaled it at some point of time and we may have not even realized. Um, so the relationships of this spider or the spider tree of life in general, our understanding has been evolving from morphology, gradually transcending to a combination of morphology and Sanger sequencing. And now, with the explosion of these genome scale data sets, we have transcriptomes, UCEs, introns, and even whole genomes. So let's take a quick recap of how our understanding has evolved and which papers actually did that recently. So these two papers had the first transcriptome-based phylogenies in the year 2014. Uh, the first paper, by the way, here uh, um, told us that or concluded that all orb-weaving spiders are not monophyletic, that cribellate and ecribellate spiders are grouped differently. And then the recent paper, well, not recent really, it was in 2017, the largest spider phylogeny with 932 spiders or terminals on that phylogeny, with all the known families at the time except one, Synaphidae, and which told us a much better picture than what we had already known. And we came up with a UC based phylogeny to see how the relationships are affected by this different data class. And then since then, our understanding continues to develop. And the most recent hypothesis about the relationships of spiders is this Kalal et al. Paper. In all these studies, the symphytognathoid spiders, or the group of miniature orb-weaving spiders, were represented. However, the question still remained uh, how to address their conflict in their relationships. So when I say small spider, it looks big on your screen, but this is just 1.5 millimeters long spider from Chile. Now, anybody who may even hate spiders or be scared of spiders will appreciate that this looks cool. Well, at least to me. So this belongs to a group called symphytognathoids, which include four families. There is a fifth one called Synaphidae, and for today's talk, this is the family of our interest with 58 genera and 233 species. Um, and this is to give you an example to make you appreciate even more to this family, because the one on these first two rows are a blind spider from Croatia found in a cave, whereas the bottom pane here shows the same species belonging to the same genus Comaroma, with, as you can see, it's highly pigmented and has several eyes. So let's take a quick look about the webs of these four families. Although these spiders may be small, there is a huge diversity of the web architectures that they construct. So this Theridiosomatidae spider uh, builds an orb web, but the hub is tucked out from away from the plane. And the another family, Mismenidae, builds this variety of modifications of the orb web. Interestingly, going back to Theridiosomatidae, there is this web uh, belonging to the built by the genus Ventilcarda, which can be found in Costa Rica, where they have these lines going, sticky lines going down and touching the water surface. Now, if you look at the bottom picture here, another spider belonging to the same family builds this web, which is quite interesting. And then Symphytognathidae builds this densely woven orb webs, and the family of our interest goes to another level by connecting to or sharing frames of orb webs, or having These kind of webs where you can see the number of radii or true radii are very small, but as you go away from the hub, you can see there are many supplementary radii. And this spider here has a more three-dimensional structure where there are some above plane radii and some additional lines going away from the plane of the orb web. Now, when we say that these spiders are small, the peculiarity is not restricted only to the webs. So in about 94% of the known spider species, approximately the ratio or the diameter of leg is close about the, let's say, one third of the diameter of the pedipalp. We are talking about the female spiders here. In Anapidae, this ratio is quite reduced, or the pedipalp diameter is quite reduced, as we can easily notice here. And in Symphylognathidae, the family of the smallest spider in the world, this is lost. When I say lost, it's a little incorrect because the first segment of the pedipalp is still present, which is an endite. And respiratory system has also some peculiarity in these groups. So across the spider tree of life, based on this recent paper and several other papers before, the two pairs of book lungs are considered as a primitive structure, as we can see them in mygalomorphs and also mesothelae, and also in some early diverging araneomorph spiders. But in Anapidae and many other araneoid families, we see a common structure where the anterior book lungs and posterior tracheal systems are present. As I already mentioned, Anapidae, this is a typical structure that the anterior book lungs are present, and if you dissect it out, these are the leaves that you can see, which are used for breathing in the spiders. But in some other anapids, we see that the anterior book lungs are replaced by tracheal systems. And tracheal systems are tubular structures that supply oxygen directly to the tissue. I think it should be this should be replaced with trachea. Sorry for that error. And in some other spiders, the posterior respiratory system is lost altogether. And here's the diversity of respiratory systems that we see across different anapid spiders. Now, to understand how the web has evolved, how the respiratory system has evolved, or how the pedipalp has evolved, we need to have a phylogeny, or the truth about their evolutionary history. But guess what? The story has not been that easy to address. Morphology and behavior and Sanger sequence data combined with morphology suggest us that they are monophyletic, that all these small spiders evolved from a common ancestor. But since we started having the molecular data, which we hope will be the silver bullet, Sanger sequence shows that they are paraphyletic, and with more molecular data with the transcriptomes, they say they are polyphyletic. So it would have been really nice if they were monophyletic, but genome scale data showed that they are not. So we tried to build another parallel set of data, genome scale data, to have a comparison with transcriptome, which was UCEs. At the time, Arachnida probe set which could target up to 1000 UCE loci was available. We designed a new probe set for spiders using the then available genomes of spiders, which could target a little bit over twice the number of UCE loci that were available at the time. So we call this the spider specific probe set. And this is a beautiful phylogeny produced using those probe set. This study was published last couple of years ago now. So let's compare what we found about the symphylognathoids. They are monophyletic. Now UCs were the first molecular data to show that all these miniature orb-weaving spiders are monophyletic. But when comparing that with transcriptomes, they show paraphyletic. Interestingly, both were highly supported, which now we judge a relationship based on the support metric. But what if conflicting relationships are highly supported? Well, it was not so easy, and I realized that I'm going way away from the questions about the respiratory system and web evolution. So to reiterate, UCs and morphology show that these spiders families are monophyletic. So the first problem was not dealing with the web or respiratory system, but to understand are they monophyletic or not. And then there are couple of three anapid genera which are found in Chile and Australasia, which begs the question, across the vast Pacific Ocean, how could these miniature orb-weaving spiders travel?
Yeah,
And how did
5 minutes here.
Thanks. How did the foraging orb web architectures in symphytic originate and diversify, and also understand the evolution of the respiratory system? So we decided to go to these places. We packed our bags and we were in a cave in Tasmania. And from there, all the way eastern and southern part of Australia, and then later to uh, Chile. So this was what the field work looked like. Although I'm talking from an air-conditioned room, this is where the real beauty of these spiders live. Since you may have guessed from my accent, I'm from India, so a civilization where thousands of years of Indian cuisine and spices have evolved. Uh, so I had to go to an Indian restaurant after a few days. Uh, Thanks to Mikel and Gustavo, Mikel Arnedo from Spain, one of our collaborators, and also came across a fake Queen band. So going from Australia to Chile, I made sure that I had enough supply this time, and these are the observations that we found on the field. So a beautiful orb web with the below plane radii and above plane radii with above plane radii on water and so on, like just beautiful diversity. Back to the lab, we also tried to address the conflicting relationships between transcriptomes and UCs by deriving amino acid data, nucleotide data from both, and then combining the regions, and then trying to look at non-coding regions and so on to see which data class gives a stable hypothesis of relationships. And in addition to the typical bootstrap, we also looked at the concordance factors, which is the proportion of sites or genes that support a particular relationships. And guess what? UCs as amino acids or as nucleotides obtain them as monophyletic. Only the amino acid data of transcriptomes recover them as polyphyletic, which is quite interesting. So I was very happy that the morphologist, the monophile is there, of course, and the morphological hypothesis still stays true. And then we would go back to the question. So Anapidae can be divided into three major clades. Vichitra clade. Vichitra means strange in Sanskrit because this is a strange clade with spiders that do not have any unifying morphology that could be called coded as a putative synapomorphy. Then microphalcomatines and all the orb weaving anapids come together form a clade. And then looking at the biogeography, these are the areas that were coded, and we found that there is an Gondwanan origin for the family Anapidae, uh, a Neotropics plus Andean origin for microphalcomatines plus the Vichitra clade, and a combination of Oriental, Neotropical, and African for the orb-weaving spiders clade. And these are the areas for the these are the combined regions which reflect Gondwanan, although at the time they looked like this. And the respiratory system, this is the coolest part of the study, where we find that although many aranioid families have anterior book lungs, there was a switch from book lungs to tracheal systems at the base of symphytic or the common branch of the symphytic families. And let's talk about the posterior respiratory system, where tracheae have lost multiple times. And the web evolution, understanding the evolution of web has been, as you may know from the literature, has been really difficult. So we do attempt here, and what we find is that there was an orb web in the beginning, or the common ancestor of symphytic, but there have been switch from orb web to non-orb web, or even loss of web, where a couple of spiders, one of them a kleptoparasite, has lost web altogether. And one more thing, a cool discovery that we found is that the web of Minanapis, which is a very small anapid, was unknown until now. And in Chile, during our fieldwork, we found that they do build an orb web, and right so it falls in the orb weaving anapid's clade, and it has above plane radii, and the diameter of this web is just 2 centimeters, and the web is built way inside the moss. As you can see, I was not very successful at getting a full picture, but this is what it looks. So with this information to you, I would hope that you will appreciate anapids, and I would like to thank all these funding agencies and Charles Griswold in particular, who during my last visit to Calacat told me that he has been collecting anapids for me since before my birth. So thanks, Charles, for all the specimens which are now on the phylogeny. Thank you.
Wow. Amazing work. Thank you so much, Siddharth. Unfortunately, we don't have time for questions at the moment, but there are a couple in the chat. Please take a look at that. So